Welcome back. Today we're going to be talking about sampling designs. Although we want to draw conclusions about statistical populations, we can't measure everything, and as a result, we need to sample. The degree to which that sample is representative of the statistical population will depend in part on whether that sample is collected in a biased or an unbiased way. What do I mean by sampling bias? Sampling bias represents the tendency for some individuals in the population to be more likely to be sampled than others. So for example, if we were interested in the attitudes of Coloradans towards environmental degradation, we would surely get a biased sample if we distributed questionnaires at a hiking trailhead or at an oil refinery. Similarly, when sampling in biological scenarios, we might find that some parts of a lake are particularly hard to access, and as a result might be less likely to appear in our statistical sample. Or other individuals or entities might be easier to find or measure. For example, you might use live trapping techniques to sample small mammals from a population. But some really nice studies have shown that more bold individuals are more likely to be captured in a trap. And some individuals might be more likely to escape handling and get free before measurement. All of these situations lead to a statistical sample of observations that is no longer representative of the statistical population that we want to draw our inferences about. Sampling bias is thought to be one of the main contributors to the collapse of the Atlantic cod fishery. Fisheries managers relied on catch estimates from commercial fishing boats as estimates of the abundance of cod in the population. The problem was, as the abundance of cod declined, the fishing boats congregated in the remaining high-density areas, and as a result, their catch didn't decline. And so the statistical sample of the entire population that came from the commercial catch numbers was biased upwards. And as a result, fisheries managers didn't understand that populations were in decline. Using commercial fisheries catch as an estimate of the abundance of wild cod is an example of a sample of convenience. That is a statistical sample that is composed of data that become available opportunistically rather than being intentionally collected by the scientist or the observer. We need to be particularly careful of samples of convenience because they have a high tendency to be biased. Another example of a sample of convenience is the magnificent eBird database, where individual volunteers report sightings of individual birds when they see them. As a result, we have a wonderful data set on observed instances of birds, but we don't have data on instances where no birds are found. As a result, the observations of where birds occur is a sample of convenience. And the folks at the Cornell lab responsible for the eBird database have gone to great lengths to try and account for the various biases that might crop up in a sample of convenience like this. So how might we then avoid sampling biases in situations where we are the ones designing our experiments or observational studies? I'm going to talk about three main sampling designs that can be used to avoid sampling bias. The first and most well-known is referred to as random sampling. In simple random sampling, all individuals in the statistical population have the same chance of being sampled, and the selection of any one particular individual must be independent of all the previous selections. So for example, if we were interested in collecting a statistical sample of the size of trees in this forest, we would need to make sure that every single tree in the forest had the exact same chance of being included in our statistical sample as any other tree. So how might we go about doing that? First, we would create a list of every individual in the population and give each individual a number between 1 and the total population size. We would then decide on the sample size for our statistical sample and use some sort of random number generator, such as the one available on the website random.org, to generate random numbers between 1 and the total number of individuals in our population to determine which individuals in the population ought to be included in our statistical sample. We would then measure those individuals whose numbers matched those produced by the random number generator, and those observations would then become our statistical sample. Now, obviously, assigning numbers to every single tree in a forest is not a practical alternative. So we need a much more straightforward solution to this problem. As an alternative, we might instead randomly pick locations, geographic locations within the forest, and sample the nearest tree to that particular location, which would also result in a random statistical sample. For practical reasons, after having measured this first tree, it might be tempting to then measure the tree right next door to avoid having to walk all the way across the forest to find another tree. But this, of course, would then not be an independent sample. The presence of this tree in our statistical sample depended on the fact that this tree was already included in the sample. And you can imagine that there might be all sorts of reasons why these two trees living right next to one another might be more similar to one another than trees living far apart. They might share the same light environment, the same nutrient environment, or perhaps the same water environment. Rather than following the procedures that I've just described for random sampling, some researchers might be tempted to just go out and measure some things. I'm just going to count the first 10 that I see. I'm going to walk through this field and just arbitrarily pick 25 flowers to measure, or I'll reach into the nest and just grab the first chick out of the nest that I can get my hands on. This is what I would refer to as arbitrary sampling or haphazard sampling. And importantly, it is not the same as random sampling. And this is because humans have subtle cognitive biases that prevent us from sampling randomly without some formal plan to guide us. We naturally tend not to select the first or last item in a list. We tend not to sample individuals from the edge of a field as much as we would from the center of the field when we follow haphazard sampling protocols like this. And as a result, haphazard sampling protocols can lead to a biased sample. So although haphazard sampling is easier than random sampling, it can lead to a biased statistical sample. So most of the time, a random sampling design is going to be your best way to get an unbiased sample. In particular, random sampling is preferred when the population is known to be fairly homogeneous, meaning consistent, or when we don't know very much about what the statistical population is like. However, there are some circumstances where we might actually prefer a sampling design that is intentionally not random. When our statistical population is made up of clear subgroups or contains an obvious environmental gradient, we might instead prefer a stratified random sampling design. In a stratified random sampling design, we divide up our statistical population into strata or groups that tend to be more homogeneous and randomly sample within each of those strata. We then combine the subjects across strata to make our overall statistical sample. So for example, if our statistical population included some areas in the forest and some areas in the field, we might use forest versus field as our two strata or groups, randomly sample within forests, randomly sample within fields, and then combine those observations together into a single statistical sample. This identification and sampling within strata as opposed to across the entire population, is designed to ensure that each of those subgroups or strata are equally represented in our final sample. And this can lead to a sample that is more representative of the population than a simple random sample would be. A second alternative to a simple random sampling design is called a systematic sampling design, where subjects are sampled at regular intervals in space or time. For example, the Alberta Biomonitoring Institute surveys biodiversity at regular 20-kilometer intervals throughout the province of Alberta. If you were interested in tracking the abundance of a species, you might also sample them at regular intervals through time, such as every month or every year. This sort of systematic sampling design is most efficient when time or space is of particular interest as a research question. In addition, systematic sampling is often much easier than random sampling, and in many cases works just as well at generating an unbiased representative sample. However, there is one important caution to be aware of with respect to systematic sampling. You want to avoid systematic sampling if you suspect that observations might vary in some sort of periodic fashion through space or time. Here is an example data set of the classic snowshoe hare and lynx cycles in the Yukon Territory of Canada. The height of the red bars represent the abundance of snowshoe hares each year between the late 1970s and about 2010. And you can see that the snowshoe hares go through periodic fluctuations in abundance. This is a situation where we would want to be very cautious about systematic sampling. Suppose, for example, that instead of sampling every year, we instead decided to sample, say, every 8 years, where these blue bars are indicated. If we were to do that, we would get a biased statistical sample that underestimated what the typical abundance was of snowshoe hares. If we did that same interval of sampling, but at a different time period, we might instead get a very upwardly biased estimate of snowshoe hares. The main point being, when there's periodic variation, either through space or through time, we want to be extremely cautious about a systematic sample because this sort of sampling design could lead to a biased sample in situations where we have periodic fluctuations in the things we're trying to sample. Even with an unbiased sampling design, we might still end up with a biased sample. This could occur through ambiguous wording to questions or misleading questions, inaccurate responses, or data that are missing from the sample in a biased way, or many other possibilities depending on the specifics of your study. The take-home message is to make sure that even though you implement an unbiased sampling design, you should still revisit your statistical sample to try and determine whether or not it's biased in some other way. So when implementing our statistical sampling, we ought to recognize that not all samples can provide valid inferences about the larger population. Some samples might in fact be biased. We need to think about the possible sources of bias and try our best to avoid them. We need to develop and implement sampling designs that are specifically designed to avoid a biased sample. But even when we implement these unbiased sampling designs, we need to revisit our sample and consider whether anything in our data collection might have prevented us from implementing our plan and potentially resulted in a biased sample. Collecting unbiased statistical samples is the key to drawing appropriate inferences about statistical populations.